Do you eat food? What a silly question. Of course you do. When you eat, your food is digested into nutrients that are imported into your cells to provide building blocks for the molecules that they need and to provide them with energy. You consume carbohydrates, lipids, proteins, and nucleic acids, which become the building blocks of your body. Therefore, the phrase that you are what you eat is actually true. Carbon is found in numerous biological molecules, including the molecules that you eat mentioned on the previous slide. Carbon is able to form bonds with many other atoms due to 4 openings in its valence shell. Because it needs 4 electrons to fill its outermost shell, it can share electrons with 4 other atoms. This leads to a wide variety of structures in carbon-containing molecules, including chains, branches, and rings. The molecules that you eat are organic molecules. Organic molecules have a carbon skeleton with hydrogen atoms attached. Besides hydrogen atoms, there are other groups of atoms that may attach to the carbon backbone called functional groups. These functional groups determine the characteristics and chemical reactivity of the molecules because it's the atoms in these functional groups that participate in chemical reactions. Here you see the functional groups found in many different biological molecules. Hydroxyl is OH. Methyl is CH3, carbonyl is C double bond O, which refers to carbon and oxygen sharing 2 pairs of electrons in a covalent bond. A carboxyl group is COOH, an amino group is NH2, a phosphate group is PO4, and a sulfhydryl group is SH. The R refers to the rest of the molecule to which the functional group is attached. The molecules that we eat are very often large molecules called polymers that are made by attaching smaller molecules called monomers together. Poly as a prefix means many, and mono as a prefix means one. An example is that monosaccharides, which are individual sugar molecules, are linked together to form starch, which is a large carbohydrate found in fruits and vegetables such as potatoes and apples. During digestion, large polymers are also broken down to yield monomers. For instance, when you eat a potato or apple, the starch gets broken down to monosaccharides to be imported into your cells. In the previous lecture, it was mentioned that water is involved in most chemical reactions in our cells. This includes dehydration synthesis and hydrolysis. During dehydration synthesis, monomers are linked together to form covalent bonds, releasing a water molecule in the process. The loss of water refers to the dehydration part of this reaction. Synthesis refers to the building part of this reaction. Hydrolysis is the exact opposite. In this case, a water molecule is added to break molecules apart. Hydro refers to water, and lysis means to cleave or break. In the dehydration synthesis reaction depicted above, 2 molecules of glucose are linked together to form the disaccharide maltose. Di as a prefix means 2, so in this case indicating 2 glucose molecules. In the process, you see that a water molecule is formed, depicted in red. In the hydrolysis reaction shown here, the disaccharide maltose is broken down to form 2 glucose monomers. Note that this reaction is the reverse of the dehydration synthesis reaction shown in figure 2.2c. Essentially, water is added back in to cleave apart these 2 molecules. The 4 organic molecules found in organisms are carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates are sugars, so they come from anything that is sweet, as well as breads, fruits, and vegetables. Lipids are obtained in the diet from fats and oils. Proteins come from meat, nuts, tofu, or egg whites, while nucleic acids, which are DNA and RNA, are found in all animals and plants since these are the genetic molecules. Foods such as bread, fruit, and cheese are rich sources of biological macromolecules. We will discuss each of the 4 organic molecules beginning with carbohydrates. Carbohydrate means carbon plus water. Carbohydrates are composed of carbon, hydrogen, and oxygen in the approximate ratio of 1 to 2 to 1, so essentially carbon and water. If we look at CH2ON, N refers to the number of carbons in the backbone, and this refers to how long the carbohydrate molecule is. Because carbon is attached to water, carbohydrates are highly water-soluble. Once again, sugars readily dissolve in water. Simple sugars are shorter carbohydrates and include monosaccharides, which are only one individual sugar molecule. Recall that mono as a prefix means one. Examples of monosaccharides are glucose, galactose, and fructose. Glucose is the most common monosaccharide and is transported through the bloodstream in humans. The function of monosaccharides is a quick energy source. Have you ever heard of the so-called sugar high? That's because sugars provide quick energy sources. Glucose, galactose, and fructose all contain 6 carbons and the same chemical formula, C6H12O6. However, notice that they have different structural arrangements of atoms. I've boxed these differences in for you. In glucose versus galactose, you can see that the 4th carbon down has the hydrogen atom either on the left or the right. In fructose, the carbonyl functional group, which is a C double bond O, is simply located further down in the molecule. Glucose, galactose, and fructose all contain 6 carbons and have the same chemical formula, C6H12O6. However, notice that they have different structural arrangements of atoms. I've boxed these differences in for you. In glucose and galactose, you can see that in the 4th carbon down, the hydrogen is either on the left or the right. In fructose, the carbonyl bond, which is the C double bond O, is located further down in the atom. The 6-carbon monosaccharide glucose exists in equilibrium between linear and ring forms. When the ring forms, the side chain closes and is locked into one of 2 slightly different forms. Fructose, which has 6 carbons, and ribose, which is another monosaccharide with 5 carbons, also form rings, although as seen here they form 5-membered rings as opposed to the 6-membered ring of glucose. Another example of a simple sugar is a disaccharide, which consists of 2 monosaccharides linked together. Examples include sucrose, which is table sugar, which consists of glucose plus fructose, maltose, which is grain sugar consisting of 2 glucose molecules bonded together, and lactose, which is milk sugar, which consists of glucose plus galactose. The function of disaccharides is short-term energy storage, because the molecules can simply be broken apart into monosaccharides to quickly release energy. Sucrose is formed when a monomer of glucose and a monomer of fructose are joined in a dehydration synthesis reaction. In the process, a water molecule shown in red is lost. Common disaccharides include maltose, which is grain sugar, lactose, which is milk sugar, and sucrose, which is table sugar. Complex carbohydrates are longer carbohydrates referred to as polysaccharides. These consist of many monosaccharides linked together. Recall that poly as a prefix means many. The first function of polysaccharides is long-term energy storage. In animals, we have glycogen, which is a polysaccharide found in our muscle and liver, which provides energy for future use. In plants, starch is found in roots, fruits, and vegetables. Amylose and amylopectin are 2 different forms of starch. Amylose is composed of unbranched chains of glucose monomers, while amylopectin is composed of branched chains of glucose monomers. Because of the way the subunits are joined, the glucose chains have a helical or coiled structure. Glycogen, which is not shown here, is similar in structure to amylopectin but is more highly branched. Another function of polysaccharides is to provide structural support. Chitin is the hard body covering found on insects, spiders, crabs, and lobsters. If you have ever eaten crab legs, you have had to crack through the hard chitin covering to get to the meat inside. Essentially, in these organisms, the skeleton is on the outside rather than on the inside. Cellulose gives rigidity and structure to plant cells. For instance, tree trunks and tree branches are rigid due to cellulose. Most animals cannot digest cellulose, therefore it is not a food source. Insects have a hard outer exoskeleton made of chitin, a type of polysaccharide. The next organic molecule that we discuss is lipids. There are two main features of lipids. First, they contain large regions of carbon and hydrogen with nonpolar covalent carbon-carbon or carbon-hydrogen bonds. It's these nonpolar regions that make lipids hydrophobic and therefore water insoluble. There are three major groups of lipids to discuss. First, triglycerides, which consist of fats, oils, and waxes. Next, steroids, and finally, phospholipids, which are found in cell membranes. We will not be covering phospholipids in this unit, but we'll save those for the next unit. First, we discuss triglycerides that contain carbon, hydrogen, and oxygen atoms. Triglycerides are formed by dehydration synthesis of 3 fatty acids plus 1 glycerol. That's where the name comes from. Tri means 3, referring to the 3 fatty acids. And glyceride, well, it kind of sounds like glycerol. The function of triglycerides is long-term energy storage in plants and animals. In humans, we have a layer of adipose tissue, which is fat stored underneath our skin. Triglycerides have a higher concentration of chemical energy than both carbohydrates and proteins, making long-term energy storage useful for this molecule. A triglyceride is formed by the joining of 3 fatty acids to a glycerol backbone in a dehydration synthesis reaction. 3 molecules of water are released in the process, 1 for each of the fatty acids. Triglycerides include saturated fats and unsaturated oils. Saturated fats come from animal sources such as bacon fat and butter, which are solid at room temperature. Saturated refers to the fact that the carbon atoms in the fatty acid tails are attached to as many hydrogen atoms as possible. Due to all of these bonds, saturated fats are harder for the body to digest, so they can clog arteries. Unsaturated oils come from plant sources such as corn and olive oil. These are liquid at room temperature. This is because there are fewer hydrogen atoms attached to the carbon in the fatty acids, allowing for fluidity. Due to less bonds for the body to break, these are easier to digest and therefore are healthier for us than saturated fat. Here you see the difference between a saturated and unsaturated fatty acid. Notice the tight packing of hydrogen atoms in the saturated fatty acid that makes the molecule more dense and therefore a solid. In the unsaturated fatty acid, you can see that there are less hydrogen atoms giving room in the middle of the molecule that allows for movement or fluidity, which is what makes this a liquid. Trans fats are artificially produced through the process of hydrogenation, meaning adding hydrogen atoms to oils to solidify them. An example would be margarine, which is essentially a solid oil. The problem is that this can produce trans fats with hydrogen atoms on opposite sides of a carbon double covalent bond. Trans means opposite or across. These are not normal bonds that our body recognizes, therefore we have an even harder time digesting these than saturated fats. You should pay attention to nutritional labels. Not only should nutritional labels give the total percentage of fat, they should also give you the breakdown of saturated, unsaturated, and trans fats as well. Saturated fatty acids have hydrocarbon chains connected by single bonds only. Unsaturated fatty acids have 1 or more double bonds. Each double bond may be in a cis or trans configuration. In the cis configuration, both hydrogens are on the same side of the hydrocarbon chain. In the trans configuration, the hydrogens are on opposite sides. Recall that triglycerides consist of fats, oils, and waxes. Waxes are chemically similar to saturated fats, except they are solid at normal outdoor temperatures. Examples of waxes include the wax in your ears, the honeycomb of a bee, and the covering of leaves. Consider the fact that if you go outside on a hot day, your earwax certainly does not melt and ooze out of your ears. However, if you were to take a saturated fat such as butter and put it outside on a hot day, it would melt. Waxy coverings on some leaves are made of lipids. Steroids are the second group of lipids, but they look nothing like triglycerides as they consist of 4 carbon rings with functional groups attached. An example of a steroid is cholesterol, which is found in egg yolks, for instance. While cholesterol does get a lot of negative attention in our diets, we do need some cholesterol in our bodies, as it is a component of animal cell membranes, and we also use it to synthesize the steroid hormones, testosterone, estrogen, and cortisol. Steroids, such as cholesterol and cortisol, are composed of 4 fused hydrocarbon rings. The third type of organic molecule to discuss is proteins. Proteins have various functions in different organisms. For instance, they function in structure, such as the keratin found in hair and nails and the collagen found in skin. They function in movement, which includes skeletal muscle in animals. They also function in transport, which includes hemoglobin and red blood cells. Oxygen binds to this hemoglobin for transport through our bloodstream. Proteins also function in energy storage. For example, albumins is essentially the egg whites in eggs, which will provide energy for the developing chick, which is the egg yolk, and there are enzymes which guide chemical reactions in all cells. Proteins consist of amino acid monomers strung together. There are 20 different amino acids found in our cells that all have the same basic structure. All amino acids have a central carbon with the following attached atoms or molecules: a hydrogen, an amino group, a carboxyl group, and a variable group. Amino acids have a central carbon to which an amino group, a carboxyl group, a hydrogen atom, and a variable group or side chain are attached. The variable group is depicted by the letter R, which essentially represents the rest of the amino acid. The R group is what differs in all 20 amino acids. Dehydration synthesis links together amino acids to form covalent peptide bonds. This yields a polypeptide that has many amino acids bonded together. Once a polypeptide is folded into its final 3-dimensional functional shape, it is referred to as a protein. Peptide bond formation is a dehydration synthesis reaction. The carboxyl group of one amino acid is linked to the amino group of the incoming amino acid. In the process, a molecule of water is released. Polypeptides fold into 3-dimensional shapes determined by the sequence of amino acids to yield a protein. There are 4 levels to protein structure as follows. Primary sequence refers to the unique sequence or order of amino acids found in each polypeptide. Secondary structure is hydrogen bonding between amino acids that causes the chain to start bending. Tertiary structure results from interactions between variable groups which causes more bending and folding. While many proteins are fully functional with tertiary structure, some proteins require 2 or more folded polypeptides joined together in order to be fully functional. In this case, this is referred to as quaternary structure. The 4 levels of protein structure can be observed in these illustrations. Primary protein structure is the sequence of a chain of amino acids. Secondary protein structure results from hydrogen bonding of the peptide backbone. Tertiary protein structure is 3-dimensional folding due to side chain or variable group interactions, while quaternary protein structure is a protein consisting of more than one amino acid chain. An example of a protein that has quaternary structure would be hemoglobin that binds to red blood cells to carry oxygen. A protein's three-dimensional shape enables it to function. Therefore, if you disrupt a protein shape, this makes it non-functional. This is referred to as denaturation. Extreme environmental conditions such as heat, ultraviolet radiation, high salt concentrations, or acidic or basic solutions can denature a protein. Some examples of protein denaturation include frying an egg. In this case, you denature the albumin in the egg white and you can physically see it change from a clear liquid to a white solid. If your fever gets too high, the reason that you die is because you start denaturing your own cellular proteins. The last type of organic molecule to discuss are nucleic acids. Finally! Nucleic acids consist of nucleotide monomers. A nucleotide has a 5-carbon sugar, deoxyribose or ribose. These are slightly different than the 6-carbon sugars, including glucose and fructose, discussed earlier in this lecture. Nucleotides also have a phosphate group, which is a functional group with 1 phosphorus atom and 4 oxygen atoms, and then also a nitrogen ring base, including cytosine, guanine, adenine, thymine, or uracil. There are 2 types of nucleic acids, deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. The sugar found in the nucleotides of DNA is deoxyribose, and those nucleotides include the bases cytosine, guanine, adenine, or thymine. DNA is referred to as the genetic molecule. RNA has the sugar ribose and one of the following bases, cytosine, guanine, adenine, or uracil, which differs from DNA that has thymine instead. RNA is copied from DNA to direct protein synthesis. A nucleotide is made up of 3 components, a 5-carbon sugar, a phosphate group, and a nitrogen ring base. Both DNA and RNA contain nucleotides. All nucleotides contain a phosphate group, but in DNA the sugar is deoxyribose, while in RNA the sugar is ribose. Each nucleotide also contains a nitrogen ring base. These nitrogen ring bases include cytosine, adenine, and guanine, which are found in nucleotides in both DNA and RNA, while thymine is found exclusively in DNA and uracil is found exclusively in RNA. DNA is a double helix consisting of 2 strands of nucleotides coiled like a corkscrew or slinky. The sugar phosphate backbone, indicated by the curvy lines, is on the outside and the bases are on the inside. Each base from one strand interacts via hydrogen bonding with a base from the opposing strand to hold 2 strands together.